So today I've got a whole bunch of substances here. They're all a bunch of macromolecules, and we're going to do a food lab today where we're going to test each of these substances, and I'll show you what a positive test looks like and what a negative test looks like for each of these things. So from I don't know if this is your right to your left. We've got A, which is water. I'm just going to add distilled water, so it's clean, right? It doesn't contain any impurities like chlorine and other stuff like that, just plain water. The one over here is protein. I'm not going to mix it with water because I think this is already a diluted solution of protein. So, but the next one over here, glucose. Okay, so I got it from our stock in the cupboard. So we got some glucose. I'm gonna bring it up to about 30 milliliters. Okay. Next we've got fructose. That's D. Looks very similar to the glucose. Right, if I put the two together, well, it's already dissolved, but they're all kind of sugary in a way. The grains are a little finer. Where's my, about 30 milliliter. It doesn't really matter, it's just the stock that we're going to use later. Sucrose or sucrose, that's pretty much just sugar, but you might remember what the chemical looks like. It's one molecule of glucose and one molecule of fructose bonded together. And then we've got here starch. And the starch looks like this. You can imagine it. It looks and it feels like cornstarch. I'm sure you guys know what cornstarch is. We might use for food. This one is a soluble starch, okay? And so these are the ones I'm going to use. I'm going to add a little bit of each into test tubes, and then I'm going to add the reagent to these test tubes to test for the presence of specific biological molecules. I hope you see I'm wearing all the proper lab gear. I've got my safety goggles on, I'm wearing gloves, and I've got an apron, a lab apron. So the reagent we're going to use to test for starch is iodine, a solution of iodine. This is what it looks like. All right, and so I've got here about, there, you guys can see, I'm gonna add about 5 drops of iodine to each of our solutions. And remember, these solutions are saturated solutions because you can see it hasn't fully it doesn't look like everything has fully dissolved, but actually that the distilled water I added is at its capacity. It cannot, it's super saturated, or sorry, it's saturated, and so it cannot dissolve any more of the solute. Okay, and so I'm going to add 5 drops of iodine to each of my test tubes. I'm going to try and get it in the middle. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. And you should see that one of these things is not like the other. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. All right, so what do we see? F changed color, right? It looks to be completely dark, and so this kind of amber color of the iodine changed to a dark blue, almost black, when I added it into the F, which is the starch solution. Another thing that contains starch that I have readily available is paper. So what I'm going to do is I'm just going to take this black paper and let's see what happens when I add some of this iodine onto the paper. It usually also gets pretty dark. There. You could see the back side of the paper. Still looks like a little bit amberish, but it's definitely darkened. It makes this dark blue, black type color. Let me go get a tissue. I've got some tissues. Let's see, that might actually work better. Okay. So just to show you that paper also contains complex carbohydrates. Wow, in this case, you know what? I think this tissue is not made of paper. This might actually be some kind of synthetic material. That's very weird. If it was toilet paper, it certainly would have changed into a dark blue. So that was a surprise even for me. Alright, ninjas, and now the test for lipids. I'm going to add some distilled water to the speaker. And what happens when I add a lipid or oil? This one is vegetable oil. What happens when I add the oil to the water? You know what's going to happen. It's going to separate, right? Just close this lid. And so since it's a vegetable oil, it's got unsaturated fatty acids, right? You can shake it. You see the separation there between the oil and the water. Okay, doesn't matter if I mix it up. It's gonna keep separating like that. Very, very simple procedure to test for lipids, and that is to add a drop of it, or add a little bit, to paper. And so I've got brown paper here. And what should happen is, once it dries, it leaves a fat stain. Same thing that happens to your clothes. And so it's going to make the paper look translucent even though the paper dried up. You know what? I'm going to put this in front of the fan so it can speed up the process of drying. And then I'm going to show you guys later. Now there's another test that we can do for lipids, and that is to add alcohol to it, and then we should get what is described as glass beads if you mix it up. So I'm going to add a few drops of ethanol. Yep, that's definitely alcohol. So we got this. The ethanol, it's pretty strong. It's a 95% denatured alcohol. I'm gonna mix it, and you're gonna see what happens. Can you see those glass beads there? That is kind of the idea. There we go. All right, next we're going to test for the presence of proteins. And so the reagent we use in that case is Biuret solution. It's made up of copper sulfate and 30% sodium hydroxide. So it's definitely not something that I want to get on my skin, or definitely I don't want to get it in my mouth, because it is at least 10 times more toxic than regular salt. Which you might not think is toxic, but it is at a certain amount or concentration. Okay, let's take a look. If I add 10 to 15 drops of Biuret solution, and it's usually about 10 drops when I squeeze the whole thing. So 1, 2, 3, 4, 5 more. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. One, two, three, four, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. Okay, about 15 drops each. Okay, you might not see anything yet, and so that's why we have to shake it. Let's mix it a little bit. And now, as you can see, absolutely nothing happened. And that will be because our protein that I used is old and denatured, it seems. And so it's not giving us a result, a positive result. Yeah, as you can see, like, maybe there's like a faint color to it. But next time I'm gonna add some yogurt, and I'm also gonna take some peanuts, and we should be able to detect the positive result for protein. So this is the difference between a lab online, a virtual lab, and then an actual practical in class. So now we're going to test for the presence of simple sugars. So those will be the monosaccharides, and the reagent in this case that we use is called Benedict's solution. Okay, this is the color of our Benedict solution, and they want us to add about 1 milliliter of Benedict solution, so that's about 20 drops. And so if I measure it out, so first into my distilled water, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So it's about 10, the whole thing. So if I just like empty the whole thing, that's 10. That's 20. 20 drops, 1 milliliter. 10, 20, 10, 20, and the same here. Now we're not done. What I have to do next is I actually have to mix it, and then I have to place it in a hot water bath for 10 minutes. So let's do this. I'm gonna change my angle for my camera, if you don't mind. So you can see the board. I've got 10 minutes ready to count down. As soon as I place it in my hot water bath, which I've got over here, I've got a beaker inside of my hot water bath, so I can keep these things safe, so they don't fall out. Okay, so I kind of have to like mix it. Again, why I wear gloves? I don't want any of this stuff to touch my skin. But we should actually do it at the same time. So first I'm just going to mix it, and then I'm going to place them quickly. All right, here we go. And count down 10 minutes. Let me show you what that looks like. Okay. So here I've got all of my different test tubes, and you can already see some some color change is happening. And so that color change is going to indicate the presence of a simple sugar, a monosaccharide. Five, 4, 3, 2, 1, 0. Nice. All right, so they should be done now. I can take them out. These are the worst test tube holders I've ever found in a lab. I hope I don't drop anything. That's A. Jeez Louise. Let's be. C. That's F. Actually, you know what? I think this is C. This one's D. Yikes. Nope, this is D and this is C. Okay, so what did we find? No, sorry about that. Okay, so here's our results. I see at least 3 colors there, right? I want you guys to describe the colors that you see. So A was just water, right? Clean water. B was the protein. C was glucose. D was fructose. E, sucrose, and F was starch. It's now 1 hour later, and this is the result of our lipid experiment. So I put it in front of the fan for an hour, and it is dry, but you can still see this spot, this oil spot, right? And it's kind of translucent. You can sort of see through it. Okay, so in this next part, we're going to take actual food items. We've got some peanuts, which I ground up over there. I've got here dry split peas, which I tried to grind up here using the mortar and pestle. Then here is syrup. As far as I know, it's corn syrup. And then this is yogurt. And I think this yogurt's very sweet, so I think there's going to be a lot of sugar in there. So we're going to do the same tests. I'm not going to test for fats though. Maybe I'll do it for the peanuts. But I'm pretty sure you guys can imagine what's going to happen if I crush some peanuts and smear it onto paper. I'm sure you can imagine that it's going to make some kind of stain that's going to stay there, right? Peanut butter, right? Lots of fats. So let's do the test. I'm going to do the same thing with iodine, testing for starch. We've got Benedict solution, testing for simple sugars. And then we've got Biuret solution, testing for proteins. Okay, so let's start with the... Oh, I said we're going to do starch first, right? Okay, so 5 drops each. Okay. So this one with the syrup, this one with the peas, this one with the peanuts, this one with the yogurt. And so at the bottom there we see some discoloration. We see some darkness there. That might be a little bit of color change. Let's look at the peanuts. Again, the peas. Yogurt. Never thought I would add iodine to yogurt. And there's just the syrup. All right, next. I've got my Benedict solution. And so I'm gonna do the same. 20 drops each. If you want to look at the colors first off, before I mix it and put it in the hot water bath. Okay, and they're gonna go in for 10 minutes, unless I already see like a color change before 10 minutes, then I'm gonna take them out a little earlier. Maybe we did it a little out of order, but this is the Biuret solution. And so Biuret solution is going in. I said 15 drops, so. And hopefully, yeah, we actually have better luck with these actual food sources than we did with the lab protein, because I think that the protein from the lab must have been in that cabinet for a number of years. Let's guess, maybe 10 years. It could, you know, it's anybody's guess how old that protein was. Let me show you what the flask looks like. So I found this in the back of one of the cabinets. I can't imagine how old it must be. So obviously it's been denatured. But we can already see here. Okay, so this is our syrup, corn syrup. The color still looks exactly the same as when I added the Biuret. This, number 2, this is our split peas. Look at that color. Okay. Protein. Oh, sorry, the peanuts. And finally the yogurt. So I'm gonna take them all out and hold them together so you can see better. There we go. All right, let's see what's going on here with the hot water bath. Okay, so this is my number 2. Number 3. Wow, look at that. That's number 1. That's my number 4. See what they do to our yogurt? All right. So again, I'm going to speed up the time, and then I'll show you the results after 10 minutes. All right, turn this baby off. Okay. All right, so time's up, and this is our results. So number 1, syrup must be a high fructose corn syrup. This one, number 2, this was the peas. Number 3, peanuts. And number 4, the yogurt, strawberry yogurt. All right, but interestingly, one more thing, just before I let you go. This was the iodine solution. So syrup, this is after the same amount of time. So 10 minutes has passed. Look at the peas. Look at the bottom of the peas there. If I shake it, the peanuts, it's kind of trippy. And finally, the yogurt. So that was pretty yuck. That's it.